This paper suggests a therapeutic strategy of drug-resistant cancer treatment, which was inspired by HIV therapy. The flowchart shows molecularly targeted therapy for HIV infection and the similar mechanism of cancer. Drug resistance is a huge obstacle in cancer treatment, but also in HIV therapy, drug resistance occurs repeatedly. However, after the patient-specific combination therapy was emerged, HIV infection rarely progressed to incurable AIDS or death. Now, HIV infection has been transformed from a uniformly fatal disease into a severe but manageable chronic condition. Therefore, the authors suggest to introduce the patient-specific combination therapy to cancer treatment. The similarity of HIV and cancer is the development of abnormal growth and loss of balance. The factors which lead the host as unbalanced state are different. However, emergence of various genetically or epigenetically distinct disease clones eventually foster drug resistance and lead failure of treatment. Now I will explain some information about the HIV therapy. More than 24 approved antiviral drugs targeting 5 different phases of the viral life cycle, viral attachment and viral maturation, for example. These drugs are used as building blocks for combination therapy. There are some reasons of quick and successful development of multiple targeted drugs against HIV. First of all, HIV biology is relatively straightforward. Viral functions are carried out by proteins, which makes them druggable. Viral load provides an accurate biomarker. And there are 3 major sources causing drug resistance. The viruses could already be resistant to certain drugs, and the emergence of drug-resistant clones as a result of random mutations could be a reason. Finally, accumulated multiple resistance mutations also cause drug resistance. Comprise 2 or more drugs from at least 2 distinct drug classes leads successful therapy. But most of all, drug combination therapy can be overcome by drug-resistant viral clones. Patient-specific resistance profiles suggested alternative combination therapy for drug-resistant HIV. To make this profile, several information are gathered and processed. Cell-based assay of resistance of the patient's viral population against drugs were measured, and the cellular phenotype of in vitro resistance correlates with clinically observed drug resistance. Also, routine profiling of patient-specific resistance against HIV drugs based on viral genotyping, and finally, computer-assessed resistance prediction has emerged as a well-validated alternative. Now let's see the conventional cancer therapy and the limitations. There are some limitations of cytotoxic drugs as constitute ideal building blocks for patient-specific combination therapies. The cytotoxic drugs have broad toxicity which can cause strong side effects. Also, they have small therapeutic window and converge on a small number of pathways. These limitations could be overcome by using molecularly targeted drugs as the building blocks. Targeted drugs modulate proteins or pathways that are specific to a molecularly defined subset of cancers. The targeted drugs modulate disease-specific mechanisms that are absent in healthy cells and increase efficacy and limit the options for cancer cells to become resistant. Also, they have a large therapeutic window, which means that as a group, they can be used simultaneously and tackle a diverse set of disease pathways. However, regardless of using targeted anti-cancer drugs, drug resistance occurs due to various reasons. Sequential treatment quickly exhausts the arsenal of applicable drugs. That's why the prediction and personalized medicine is important. Cellular resistance phenotypes and high-throughput genome profiling are needed to devise patient-specific combination therapies. Cellular resistance phenotypes may provide a powerful approach for predicting clinical drug resistance. It will be used to predict sensitivity and resistance of single drugs and drug combinations. Carefully designed cell transplantation assay can provide patient-specific predictions of clinical drug resistance. But this approach requires high cost and labor intensity. High-throughput genomic profiling is a computational method which can be used to predict disease-specific drug sensitivities by using epigenome or transcriptome of individual cancer samples. It might attribute to obtain fingerprint or scorecard of drug resistance. By using these tools, genotype-phenotype prediction could possibly suggest patient-specific treatment. As I briefly mentioned in the intro, the current goal of the cancer therapy is shifting goals as converting a deadly disease into a chronic condition with long-term survival. To follow the successful patient-specific therapy of HIV, combination therapies involving several drugs with different mechanisms of action are needed. To achieve this, larger range of new drugs that target alternative cancer pathways and accurate biomarkers for predicting drug resistance and the toxicity of combination therapies are necessary. The genotype-based predictions of cellular drug resistance phenotypes could provide a relatively straightforward path towards personalized medicine. They suggested a two-phase approach for establishing the rational selection of patient-specific combination therapies. At phase 1, resource-intensive cellular phenotype assays are applied to a well-defined and very small patient cohort. And then at phase 2, data gathered in the first phase are used to create computational models. When the second phase is completely established, drug resistance will be predictable from genomic data of the patients and the viral information in the patient. The drug resistance problem in HIV was successfully tackled by introducing combination therapies. Building blocks of personalized medicine in HIV might be informative for shaping the future of personalized cancer therapy. By increasing number of targeted cancer drugs and emerging of high-throughput sequencing, personalized cancer therapies could be devised as HIV therapy.